Hi, I'm Emily, and I'll be presenting on our lab's most recent work in which we profiled the epigenome and the transcriptome at the single cell level in postmortem human brain tissue samples from Alzheimer's disease and controls in order to elucidate the gene regulatory programs that may be underlying neurodegeneration. And so specifically, we took human prefrontal cortex samples and we divided these in order to perform both bulk RNA sequencing and single nucleus RNA sequencing in addition to single nucleus attack sequencing on these same samples. And for those who are not familiar with attack sequencing, basically with attack sequencing, we're able to profile regions of open chromatin. And to date, there have been very few attack sequencing studies performed on the human brain, but even more importantly, there have been no previous attack sequencing studies performed on human Alzheimer's disease brain samples. And so as you can see here with both sequencing methods, we are able to capture all the major cell types of the human brain. And with single nucleus attack sequencing, we profiled around 130,000 nuclei. And in single nucleus RNA sequencing, it was around 61,000 nuclei. And so we classified our nuclei based upon the chromatin accessibility or the gene expression of these known marker genes. And in addition, while there have been a slew of genetic studies of Alzheimer's disease, and these have identified these AD-associated single nucleotide polymorphisms, we still really have a poor understanding of the functional significance of these AD-GWAS SNPs. And so we wanted to leverage our attack sequencing dataset in order to help clarify these AD-GWAS SNPs. And so as you can see here, we found significant enrichment in our two microglial clusters of AD-GWAS SNPs, and this supports previous literature. And so we also looked at the chromatin accessibility of AD-GWAS loci across the major cell type as well as at our cell type cluster level. And so this effectively creates a map of the cell types that an AD-GWAS locus is accessible in. And so we also constructed cis-regulatory networks at AD-GWAS loci. And as you can see here, with both, we were able to find both cell type specific cis-regulatory connections in addition to disease specific cis-regulatory connections. And so because we had both our epigenetic and transcriptomic information for these samples, we were able to directly integrate the two data sets, and then we performed trajectory analysis, identifying cell states that were associated with the disease that were epigenetically and transcriptionally distinct. And so here we're just showing our microglia and astrocyte analysis. But as you can see here, as you progress along this microglial trajectory, you see an increase in the AD nuclei in comparison to controls. And so we termed these at the end of the trajectory as disease-associated microglia. And we also took genes that were found in disease-associated microglia in the mouse. And we looked at these across the trajectory, and you can see here that we see an increase in these disease-associated microglia genes at the end of the trajectory. And we also extensively examined key transcription factors along the microglial trajectory, and here we're just showing SPI1 as an example. We also found distinct patterns in differentially accessible regions and differentially expressed genes along the microglial trajectory. And so we performed similar analyses in the astrocytes, not all shown here, but basically we were able to identify astrocytes that were disease associated and again, epigenetically and transcriptionally distinct. And so in addition, we also here present a new analysis method to implement gene co-expression network analysis to single cell data. And this is so that we can identify cell type specific disease associated gene co-expression modules, or in other words, groups of co-expressed genes. And here we're just showing 4 of the microglial modules which were significantly associated with AD. And so to summarize, our study reveals the cell type specific epigenetic and transcriptional changes that are occurring in Alzheimer's disease. We identify the cis-regulatory network or landscape at AD-GWAS loci by individual cell types. And we also directly integrate our 2 single nucleus data sets in order to highlight the cellular heterogeneity in glial cells in AD. And we also highlight how dynamic gene regulation is in disease. We've also made our data sets really easily accessible for any other researchers to explore. So for those who are interested, please go to our lab's website and you can easily look up any genes that you may be interested in. Thank you.